Okay, so topic 4.3 talks a little bit about signal transduction, although I don't think that's necessarily, uh, you know, the best title for this topic per se. But it wants, there's a few different things, and I actually wrote the learning objectives up here so we could discuss them a little bit. It wants me to describe the role of environment in eliciting a cellular response. Signal transduction pathways influence how the cell responds to its environment, and this is very true. These are yeast here, right? And I do want you to understand this basic process right here because we are going to witness this in the classroom. We are going to have an experiment with yeast. Um, this is a haploid cell here. This is a haploid yeast cell here, and they have a very interesting way of kind of having sex here. They release these kind of communication factors here, and there's these are very awkwardly named, right? There's kind of an A, excuse me, an A factor down here, and then this alpha factor up here, which is kind of looks very similar, right? But you have two haploid cells. Again, this means you have half the DNA of a normal diploid cell, right? And they form this thing called a shmoo. They approach each other and they form a shmoo, and they kind of combine cytoplasm, and they combine into a new diploid cell. Of course, your environment in this case contains different factors, which is going to cause, if you have a high concentration of this factor near one of these cells, it's going to bond with these receptors, right, and cause it to grow in a certain direction. Likewise, this A factor, it's going to bond over here to these receptors if there's a high concentration, and it's this kind of reciprocal nature of these signals is going to cause them to grow towards each other, and in the process, before it they form together like this, the actual shmoo is when you have one cell maybe, which we should see in our experiment. Hopefully, you'll have one cell over here, and then you'll have another yeast cell over here, and they'll start kind of elongating towards each other, right? Based purely on these signals that they are putting out, right? These signals that these are putting out in this reciprocal nature of this signaling system here. Okay, another look at signal transduction. I wanted to find kind of the most confusing, you know, pathway I could find here, just so we could see how complex these cellular responses are. So describe the different types of cellular responses elicited by the signal transduction pathway. Signal transduction may result in changes in gene expression and cell function, which may lead to apoptosis. Apoptosis, of course, is programmed cell death. Hugely important, right, in your body. You need to be able to self-destruct cells that are cancerous or have something going on with them that is not that shouldn't be. You need a way to get rid of cells that have invading pathogens or something in them. So it's very important to be able to trigger apoptosis in a cell. Of course, if you guys probably know this, but cancer, which is uncontrolled cell division. You have a problem with this, right? Apoptosis is not something that really occurs. So we have this very complex system here. This cyclin, I would like you to remember this cyclin and this cyclin CDK complex down here, because we'll revisit this when we talk a little bit about cyclins and we talk about mitosis, not long actually. Um, so I do want you to remember some of this stuff, but in general, all these different little proteins and phosphorylation that happens, you don't need to know all that. One thing I do want you to definitely know is how these pathways look and what an inhibitory pathway looks like and what an activating pathway looks like. So, an activating pathway is something in which, you know, thing X happens over here, some sort of phosphorylation usually, and this triggers Y to happen right over here. This is an activating pathway. This is an activating interaction. Of course, you have these other types of interactions in which you don't have arrows. You have kind of this blunt stop right here. This is an inhibitory action. So in other words, if you have a lot of this GSK, it's going to inhibit this further process of cyclin CDK complex formation down here. It's going to stop this process. You can think of it as having a stop sign or an X down here. This is an inhibitory process. Here's another one over here. right? So one thing I want you to notice about this is you still have kind of a ligand receptor binding point over here, right? EGF would be your ligand in this case. And notice that all these P's you see. How many P's do you see in this whole process over here? You see quite a bit of them, right? You see quite a bit of P's. These are all phosphates. Phosphorylation, you remember that word we talked about a video or two ago? Phosphorylation is huge in cell signaling. A lot of things are getting phosphorylated, which is causing chain reactions of other things to get phosphorylated, which is causing more chain reactions of things. So these phosphates are kind of being passed around um, uh, to kind of start this process. Now, this is actually the cyclin CDK complex deals with mitosis, right? And this G1S progression is something that will continue. If it continues unchecked, this is a cancerous cell, right? So this is a very well studied kind of cell signaling pathway here. The reason this is well studied is because cancer is one of those things that affects a lot of people, and scientists are constantly trying to figure out ways to kind of disrupt this process, to disrupt cancer cells without affecting other cells around there. Of course, this is the nucleus in here. You guys probably noticed that because of the DNA down here. Um, but don't get wrapped up in this. Don't draw this whole thing. If you're drawing this whole thing, please stop. You don't need to draw this. This is just I wanted to show you how complex some of these processes can get in the actual cytoplasm when you start looking at things and how they activate certain things and how some things can inhibit other processes, right? It's important to know that too. A lot of processes, mitosis is one of those, would continue unchecked if not for things that were inhibiting it, were inhibiting this process right here. Um, so just keep that in mind that there is apoptosis is a thing that is pretty normal for cells sometimes, and they sometimes need a signal for that. Gene expression, of course, the making of mRNAs here and the expression of genes into proteins, which is something we haven't talked about quite yet, but we will talk about later this year. Um, uh, is part of this whole signal transduction pathway. So again, don't get too caught up in how confusing this picture is. Just marvel at how complex that our cellular machinery can be, especially when it comes to this transduction process, which is really all of this stuff in here is all transduction. So I'll leave it at this, but before I go, I would like you to write down the 3 steps in a signal transduction pathway on your notes right now. Okay, I'll leave it at that. Write the 3 steps down, and you're done with these notes. Thank you.